methylotropes. So based on our organism, the carotenoid pathway in methylotrope is works like this. Methylotrope, they can use methanol as a carbon source. So using this methanol, it will enter the serine cycle, and from there through the MEP pathway, the natural carotenoid production pathway is there. So in its native environment, our methylotrope, they produce this C30 carotenoid from FPP and then diverted towards the squalene pathway and then produces lycopene or diapolycopene, which is a C30 carotenoid. Since we don't know the value and the uses of these C30 carotenoids, we wanted to produce C40 carotenoids in this organism. So we blocked the C30 carotenoid pathway by deleting some genes, and then we try to introduce the C40 carotenoid pathway genes. But till now, we have had some problems with this C40 carotenoid pathway. When we try to introduce in our organism, they were found to have very toxic effects. So in order to overcome that toxicity, we have been in search for some these promoters that can be controlled, that can control the expression of the C40 carotenoid genes. So establishing an inducible promoter system for expression of carotenoid genes in methylotrope. So recently we come across one promoter that is a PO promoter, which we call as PO. It's a phenol inducible promoter, and it was reported to work very well in methanotrope. And since methanotrope and methylotrope, they are quite related organisms, we wanted to try this phenol inducible promoter in our host. So we first tested this phenol inducible promoter in E. coli. In our previous the plasmid that we have been using for methylotrope, that is PMEV, we removed the PMAXF promoter and then replaced it with PO, the phenol inducible promoter, and then clone in SFGFP. And in E. coli, this phenol inducible promoter also works very well. The growth, we tried inducing phenol concentrations of 0, 5, 10, 20, 40, and 80 micromolar. And in all of these concentrations, the E. coli, there was no significant difference in their growth, and that is the phenol concentration is too low to exert any toxic effects on E. coli. But if we see the fluorescence of the SFGFP at 0, the fluorescence is almost negligible, and then at 5, 10, 20, 40, and 80, it keeps on increasing. At 24 hours, we see that the concentrations of 40 micromolar and 80 micromolar, this seems to be the optimal for E. coli expression. We have not checked higher range, but if anybody who is interested in working with this PO phenol inducible promoter in E. coli system, they can check further. But according to our experiment, we conclude that maybe 40 micromolar might be optimum for expression in E. coli. So after this test, we again move on to test in our host, that is M. organophyllum. And for this expression, we use an M. organophyllum strain delta C, delta SPNC, which is a colorless strain, in which we have removed the carotenoid production pathway in this organism. So in here, we use the same construct, PMEV, PO, with SFGFP. And here we tested 0, 20, 40, 50, and 100 micromolar concentrations of phenol. There we find that there was not much difference, significant difference in the growth when we expressed this, when we did these concentrations of induction. And at around 80, it shows a little bit lower growth, but at 100, it was okay. So maybe there, this was not because of the toxicity or maybe some other reasons. However, the pH drop and everything was, there was no significant difference. But when we see the fluorescence expression and the fluorescence of SFGFP, at 24 hours, there was not much fluorescence. because the cells were still in the lag phase. But after 24 hours, when they started to grow at 48 hours, the expression of SHGFP increased. And we found that at 48 hours, the 20 micromolar and 40 micromolar seems to show the highest expression of SHGFP. And at 72, where we terminate our growth, still the 20 and 40 seems to show the highest expression. And during this time, we consider that maybe already they have reached the saturation, and the inducer saturation already was achieved at 20 micromolar in our host, because at 40 still the increase is not that much high. So we wanted to use this 20 micromolar and as well as 40 micromolar concentrations in order to check the expression of our toxic carotenoid genes in methylotrope. So we clone in with this MEV phenol inducible promoter. We clone in our genes CRTE and B, which were codon optimized for methylotrope, along with IDI and DXS. And then when we transform this, till now, this CRTE, B, IDI, DXS, we have not been able to transform in our host because of the toxicity effect. When we express with the constitutive promoter, they were not able to form colonies. But with this phenol inducible promoter, we found that we can get a regular sized colonies with 0 micromolar phenol on the plate, with no phenol on our choricanamycin plate. However, when we also tried to check if they have still the toxicity was there with plating in 40 micromolar phenol plate also, we checked. But in 40 micromolar phenol plate, they were not able to form proper colonies. But however, due to the tight control system of this inducer, of this promoter, we were able to get this normal size colonies after 3 days of transformation using this promoter. So we tried to take, after we have got, so in here we have known that 0-hour induction still causes this toxic effect of our carotenoid genes. So we try to grow and check at different hours of induction. So this strain harboring the CRTE, B, IDI, and DXS, we grow, and then after growing, we tried 3 different stages of induction. One was induced at 0 hour, second induced at 12 hour, and the third induced at 24 hour. And then all of them grew until 48 hours. And also we used 2 different concentrations of phenol, 20 and 40, based on our previous experiment where 20 and 40 seems to perform the best. So 20 micromolar induction at 0, 12, and 24, and 40 micromolar induction at 0, 12, and 24. There were some little bit differences in the growth, especially in 20 micromolar induction. The one which was induced at 0 hour, there was the cell growth was the lowest. And we also observed that these cells, they started clumping in all the stages of our sampling. Same was almost the same case was observed in case of 40 micromolar also, but the cell seems to have sliced or something. So the OD, we don't see the OD that much difference, as a significant difference as that we observed in the 20 micromolar phenol induction. So when it comes to the carotenoid production, we have the cells we harvested after 72 hours, 48 hours of culture. The cells after, and we saw in the pellet, this is the pellet for the cells, 1 ml of cell after pelleting, which was sampled after 48 hours. So in no induction case, when there was no phenol was added, still there is the PO or phenol inducible promoter shows a leaky expression, very slight leaky expression. So the cells which are supposed to be white, they gain some little bit of color. But at 0 hour induction, in case of 0 hour induction with 20 as well as 40, we saw the pellet size was smaller because the cells were not able to grow properly, and they were having this toxic effect of the carotenoids. However, in 12 and 24-hour induction, we find that the pellet sizes were almost similar to no induction, and the color development was also slightly better. So from here, this is the first time we have been able to produce C40 carotenoids in our host with the use of this inducible promoter. So from here, we just wanted to extract. Till now, we have not optimized carotenoid extraction protocol in our host, since it is quite different from E. coli. We just wanted to try. First, we tried acetone extraction, the 48 hours culture, which was induced to 12 micromolar phenol induction, and after 24 hours of cultivation. So we extracted the carotenoid and tried to see if the produced carotenoid, the color that we saw, is either C30 or a C40 carotenoid, and if it is really a lycopene. So we run at 2 wavelengths, first for phytoin, then we saw a very big peak of phytoin and a smaller peak of lycopene. So this retention time matches exactly with the standard lycopene and with the standard phytoin. So this, the red peak indicates the lycopene and the blue peak indicates phytoin. Here we observe that a large amount of phytoin is accumulated and has not been able to convert into fully into lycopene. This is because of our construct that we introduced. Because PMEV PO, we have introduced CRTE and B alone with IDI and DXS. So it is meant for producing phytoin, but the amount of lycopene that we are getting is from the activity of the native CRTN. In this organism, although we have blocked the carotenoid production pathway, C30 carotenoid production pathway, the C30 carotenoid or phytoin desaturase, that is CRTN, is still active. But and we have also established that this CRTN is able to perform convert phytoin, the C40 phytoin, into lycopene. So from the chromosomal CRTN, since it is a single copy, it was not able to convert the whole phytoin that was produced from our plasmid into lycopene. That's why we are seeing this large peak of phytoin. So right now we are trying to optimize the carotenoid extraction protocol for our methylotrope, but till now we tried several methods: acetone extraction, BPER lysis, and then acetone extraction, and we are yet to confirm which extraction protocol will be best for our host. So ongoing work for this part is optimization of carotenoid extraction protocol in methylotropes, and then carotenoid production test. We are going to do this production test in with 20 micromolar induction, and after 12 hours, which we find was the optimal. And then with using higher percentage of methanol, that is in order to support higher cell growth. All the previous works we have done with just 0.5% methanol, but for this production test, for the proper production test, we will be testing with 1% methanol. So moving on, next, another work that we are working on was simultaneously in parallel work. We also try to elevate this carotenoid toxicity by fusion protein expression, because in some papers, many people have reported that the fusion of carotenoid proteins can help increase in the carotenoid production. So we wanted to fuse CRTI or CRTN, that is the native carotenoid desaturase, with CRTB. So we made 4 constructs: CRTE along with B and I fusion and IDI-DXS, and then CRTE with B and N fusion, and CRTI and B fusion, and N and B fusion. And for this, we chose this GGS linker with 20 amino acids, a long and a flexible linker. And one of the reasons why we choose these long and flexible linkers was that we have a little skepticism towards this fusion protein, because eukaryotic phytoin desaturase has been reported to have a tetramer structure. So a tetramer structure in case of eukaryotes, and for bacterial phytoin desaturase, the exact data have not been in our knowledge. It is not there out there still, but according to some group, they reported that it will be at least a dimer in order to accommodate the phytoin, a C40 40 carbon chain phytoin. They need to have at least a dimer. So if our protein CRTI or CRTN is a dimer, then for its function it needs to be a dimer. Then in that case, we might need we thought that maybe this long and flexible linker might be more suitable compared to other shorter or rigid linkers. So in the result, we have fused BI, IB, BN, and NB genes, and this is the first. All of these constructs were made in this constitutive promoter, our plasmid harboring the constitutive promoter. So first, BI, IDIDXS, which we try to check if normal colonies were formed. In all cases, BI fusion, IB fusion, BN, and NB fusion, they form all normal colonies, except for NB, there was a little bit slower growth in comparison to others. But when we introduced CRTE, at that time, the E, BI, IB, they were able to form colonies with lycopene production and very good color development. But in case of BN and NB fusion, we were not able to see any colonies or proper colonies or any color development. So we assume that maybe still the CRT, BN, and NB fusion, there is some problems, and the toxicity is still prevalent. But in case of BI and IB, we believe we have elevated that problem of toxicity. So right now we have we are trying to do the production test in these two constructs, that is MEV2 with CRTE, IB, IDIDXS, and EBI, IDIDXS. That much is for right now for the carotenoid project in methylotrop. And at present, we are also dealing with another project, which is antibiotic-free system in carotenoid for carotenoid expression in E. coli. This work I'm working along with Yun Che-an, our lab mate. So we first try to, in order to make the antibiotic-free plasmid system, we first try to transform this, make the PSCS2, that is the plasmid harboring the mevalonate pathway. We remove the chloramphenicol selection marker in this plasmid and then introduce DXR instead of that chloramphenicol marker. And then along with this plasmid, we mix PT-LYCM4, since this plasmid cannot survive alone. So we mix PT-LYCM4 with ampicillin marker and then transform into DH5 alpha competent cells and then plated them on phosmidomycin. So phosmidomycin addition, it will block the MEP pathway. So if the organism, if our E. coli, the DH5 alpha, if in presence of phosmidomycin, they should not be able to survive unless this PSCS with DXR is functional. If this mevalonate pathway is functional, in that case, the organism, our DH5 alpha will be able to survive and phosmidomycin containing plate. So after that, we got some, we picked up the pink colonies and grew them on 2YT ampicillin with phosmidomycin and 2YT chloramphenicol with phosmidomycin. Its colony was grown in two different types of media. In this 2YT ampicillin, our organism must be able to grow because of the presence of ampicillin marker in LYCM4. But in the same that same colony in 2YT chloramphenicol with phosmidomycin, they should not be able to grow since we have removed the chloramphenicol marker from PSCS2. And likely we got this in ampicillin, we were able to see the color development and growth, whereas in the chloramphenicol phosmidomycin, the cells were unable to grow. From here, we extracted this plasmid and then confirmed that the plasmid that we got has DXR has been removed. the chloramphenicol has been removed and replaced with DXR through a PCR, and we easily see here in A, the one which our plasmid extract, the size is bigger than compared to B, in which the this is the original PSCS2 with chloramphenicol. So after that, our final aim is to make a phytofluin plasmid with antibiotic marker-free plasmid along with PSCS. So this time, that extracted plasmid with PSCS, chloramphenicol negative PSCS with DXR, is mixed with PTLYCM4 ampicillin marker positive. So we mix this with another, our last final plasmid, that is PT phytofluin RTI SYN ligated product, and during this ligation we have already removed the ampicillin marker in this plasmid and then ligation, and after that we follow the same protocol which was used to create this chloramphenicol negative ampicillin positive strain. So from here, after plating on phosmidomycin plate, we select the whitest or white or yellowish color colonies picked up and then grow on phosmidomycin ampicillin and phosmidomycin 2YG with only phosmidomycin. In here, the colony should not be able to grow on this media because of the presence of ampicillin antibiotic, because the cells harboring these two plasmid, this PSCS2 with chloramphenicol removed and PT phytofluin ampicillin removed, so they should not be able to grow on any media containing any type of antibiotic except phosmidomycin. So in phosmidomycin containing media, they were able to grow, but when ampicillin is added, they were, and the same colony was not able to grow on this media. So from here, we extracted again the we chose 4 colonies from this from the plate from the result of this transformation, and those 4 colonies we extracted the plasmid and linearized it to see the plasmid composition. The first band is for PSCS2 without chloramphenicol, and the second band is our PTF CRTI, that is phytofluin plasmid. In all four colonies, we saw all the plasmids were in a very good condition as well as in a very good concentration. And after that, we digested, after linearizing, we again digested with XBAR1 and SAL1 to confirm further. In here, XBAR1 and SAL1 digestion, the PSCS will give 2 bands, one the biggest band and another at 1.5. As we see here, these 2 bands belong to PSCS2, and for PT phytofluin, this the middle 2 bands belongs to the PT phytofluin, and this 2 bands also confirms that the BLA or our ampicillin marker has been removed, because if the ampicillin marker was present, this 3rd band will be very close to the 2nd band and cannot be separated on a gel like here. This is the original PT phytofluin. When digested with XBAR1 and SAL1, we cannot separate the 2 bands, but in case of if we have removed the ampicillin marker, at that time these 2 bands can be separated and seen clearly like in our gel. Also, we confirm with PCR using primers that was specific for the part of ampicillin. So in the ampicillin positive PT phytofluin, we will see a band around 1.5, a little bit bigger than 1.5 kb. And if the ampicillin marker has been removed, then we will get a band around 600 base pair. So in all 4 of them, we already confirmed that this the marker, both the plasmids are marker free. So the remaining PSCS 1 and 3, right now we have following the same protocol, we have already acquired the plasmids for PSCS1 DXR with chloramphenicol removed and DXR with PSCS3 DXR with chloramphenicol removed. And ongoing is the construction of this PSCS1 and 3 along with the PT phytofluin BLA. Already we have got colonies, and the growth of this culture of these 2 plasmid mixtures are on the way. So by tomorrow, we should be able to get these plasmids ready. And the next step that we are following is to delete DXR in this MZ1655 ATCC strand. So we hope to finish this work very soon. And that much is all.
Okay, any question? 조변 교수님 혹시 질문 있으세요?
So you used the phenol inducible promoter
Yes, Professor.
in both E. coli and Bacillus.
Yes, Professor.
So is there any effector or regulator for the control of the phenol inducible promoter in E. coli?
In E. coli,
so what is the origin of the phenol inducible promoter?
phenol inducible promoter, there is another, the plasmid, okay, I'm sorry, but actually I have done this presentation in our lab seminar, so I forgot to put the whole construction and the part, but it has its own it comes with its own repressure. So the repressure is relieved with this phenol. So we use the promoter along with the repressure. We clone the repressure and the promoter in this our vector.
Okay.
Any other questions?
Shane, what's the reason it's difficult to extract the your strain using just using the acetone?
Acetone extraction. The lysis is the main problem, Dr. Kang, because unlike E. coli, our cells are a little bit complex. The cell wall structure is a little complex, so the lysis is we are not able to get full lyse of all the cells. So we are trying other different methods like chloroform and dichloromethane extraction, and they seem to have better effect than acetone.
Did you try to use the lysozyme?
Lysozyme, some people have also used, but we believe the other methods we are trying now, this two doesn't seem to work. So the chloroform and the dichloromethane extraction, it seems to be more efficient. So, but we have yet to test in some higher cell level, cell accumulated cell pellets. We have tested in some very small cell pellets. They work pretty well. So because the we think if we start extraction with the lysozyme, yes, it will be effective, but it may be a longer process compared to other extractions.
Okay.
채연아, 윤채연, 채연아, 출석을 한번 불러볼까? 윤채연, 없어? 나중에 교수님하고 계실 때 제가 불시에 출석 한번 부를 거예요. 제가 옛날에 한번 화장실 갔다 오는 거 보니까 상당히 많은 사람들이 오케이 알았어. 마이크가 안 되면 마이크가 왜 안 되지? 마이크가 셋업이 안 됐나? 오케이, sing
Yes, Professor.
So are you working with Cheon for the Macris work?
Yes, Professor.
Okay. So hopefully we can get the other two strains as well.
Yeah, we have already cultured. We have
Okay.
already got
Okay.
confirmed. Only
Okay.
we are just waiting for the culture to extract plasmid.
Okay. And then confirmed by mapping and PCR, right?
Yes, PCR. Yes, Professor.
정 교수님,
네.
저희 한번 이거 지금 만들어진 거 갖고
음.
누가 한번 컬처에서 그 한번 찍어보는 거 어떨까요? 뭐 나오고 있는지.
이거
왜냐면 그때 막 뭐지 그 안티바이오틱 있을 때는 저희가 파이토플루엔을 생산을 못했잖아요.
이거를 누가 하면 좋을까요, 그러면?
포항 이번에 뭐 하니까 같이 한번 키워보라고 할까요?
그럼 포옹이 하면 저도 찬성합니다. 예, 이거 포옹이 하면은 좋을 것 같아요. 예.
포항?
그다음
We can talk about the extraction of the phytofluent from the strain the thing got tomorrow. 제가 이거 내일 한번 그러면 저 포옹이랑 말해서 한번 키워보도록 할게요.
그러시고 좋습니다. 됐어. 저는 뭐 더 이상 없고 보자. 이게 마이크 안 되는 애들이 많네요. 왜 마이크 안 되는 애들이 이렇게 많지? 그때 캠을
그게 다 없어요. 저희 아마 그게 다 없는 것 같아요.
어떤 게 없고요?
사주면 안 돼요? 싼 거.
네? 싼 거 캠 다 사줬는데.
그 명컴에 있는 건 많이 비싸대요.
인터넷에서 한 뭐 1, 2만 원짜리도 팔거든요.
아니요, 캠 다 사줬어요.
아, 그래요?
네.
근데 마이크가 되게
마이크는
하는
보통 마이크
노트북 아니면은 따로 그
캠에 또 마이크 없나?
헤드셋을 따로 해야 해서.
헤드셋 당연히 해야지. 같이 있으니까, 그죠?
그 헤드셋도 되게 싼 거 많이 있던데.
명컴이 비싸대요.
네?
오케이, 알았어요. 그러면 헤드셋을 누가 수요 조사 좀 해줄 수 있을까요? 그러면 실태 조사 유경 쌤이 해주시면 되려나? 유경 쌤 오늘 없죠, 여기, 그죠?
우리 방장님께서 해주시면 안 될까?
손영? 손영?
네, 박사님.
그거 조사.
네, 한번 찾아보겠습니다.
오케이.
응.
아니, 조사. 누가 없는지.
마이크 없는 사람. 마이크 없는 사람 조사해 달라고요.
네, 알겠습니다.
네, 헤드셋 이런 거.
네,
캠하고.
네, 캠도 같이 조사하겠습니다.
또 다른 의견 있으세요? 없어요? 조병 교수님 고생하셨습니다.
고맙습니다.
끝났나요?
예, 오늘 끝입니다. 예, 혹시 뭐 공지사항 있으세요? 없나요?
없습니다.
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 알겠습니다. Thank you, 쌤.
Thank you, Professor.
오늘 여기서 끝낼게요. Thank you,
네,
everybody.
감사합니다.
예, 좋은 주말. 예.